Assalamu alaikum and welcome back to my YouTube channel Studyholic. Let's begin with our today's topic, that is cytoskeleton. So, I'll be briefly discussing what is cytoskeleton, a fibrous structure that is distributed from the nucleus to the plasma membrane throughout the cytoplasm of the eukaryotic cell is known as cytoskeleton. As I can draw over here, this is the cell, this is the nucleus, and the fibrous structure that spans from the nucleus till the plasma membrane is known as cytoskeleton. Now let's discuss about that fibrous structure. There are three kinds of fibers in the cytoskeleton, that are microfilaments, microtubules, and intermediate filaments. I'll be discussing them in the detail. Let's start with our first discussion, that is microfilaments. Here I have attached the diagram from the book and I'll be explaining it in the detail. So basically the microfilaments are also known as actin filaments. They are thin contractile fibers and they are about 7 nanometer in diameter. So let's discuss about its structure. It basically consists of four twisted chain. If I open up this diagram, here these are the two chains and then the beaded ones make up these all four, and these four chains have name that make up the two actin chains and then two chains of tropomyosin. The beaded ones are known as actin chains, whereas the straight ones are known as tropomyosin. And the tropomyosin chain has a triplet of troponin on it that I can show you over here. This, if I zoom it, this is the chain and this makes up the triplet on the intervals, right? So that was about the structure. Now let's discuss about the specific function of the microfilament that microfilament performs. Microfilaments are basically found in muscles, and in muscle they are responsible for the formation of the myofibrils. And the myofibrils in the muscles, which are formed by the microfilaments, are responsible for the muscle contraction and relaxation. The microfilament also performs cyclosis. So that are the two main functions. Remind them, these are the important ones. Now let's move on to the next discussion, next cytoskeleton, that is microtubule. We have discussed about the microfilaments. I'm moving towards the microtubules. So here is the diagram of the microtubules I have attached over here and I'll be discussing through it. So the microtubules are basically hollow cylinders and they are about 25 nanometers. Hollow cylinders. Okay. So they are about 25 nanometer in diameter and about 0.2 to 25 micrometer in length. 0.2 to 25 micrometer in length. I hope you get it. So if we talk about of which material the microtubules are formed, they are formed of the protein that is known as tubulin, as you can see over here. And each tubulin is dimer, that means two. As you can see, here is the blue and red color, and in the structure there are also the blue and red dimers forming the microtubules. So that's the structure of microtubule. It is a hollow cylinder kind. According to its length, it is 0.2 to 20 micrometer, and according to its diameter, it is 25 nanometer, and it is formed with a protein that is known as tubulin, and tubulin is always in the dimer, and that is indicated in this diagram by the blue and red beads. Now let's move on to the function of the microtubules. The function of microtubules have been divided among the plant cells and animal cells. In the plant cells, the microtubules are responsible for the formation of spindle fibers. How does this takes place? Before the cell division, the microtubules are freely dispersed. But at the time of the cell division, they organize themselves, and as a result, they form the spindle fibers. Here I have drawn the spindle fibers that run towards the cytoplasm during the cell division, and as a result, the chromosomes are arranged in this phase. So I'll be discussing this in further when we're discussing about the mitosis and the meiosis. Now let's forget about it. Now discussing about its role in the animal cells, what it is responsible for. In the animal cells, it is basically responsible for the formation of centrioles, cilia, flagella, and basal body. So that was about the microfilaments and microtubules. I have discussed about this structure separately, their specifications, their functions. Now I'm going to move towards the last one, that is intermediate filaments. I try to keep my lectures as small as I could, and I make them solely from the book so that you can open the book and take it, imbibe the concepts directly. Now let's move on to the intermediate filaments. So if we talk about the diameter, the microfilaments were 7 nanometer, the microtubules were 25 nanometer, whereas the intermediate filaments, they are about 8 to 10 nanometer. So this diameter indicates that it is between the microfilaments and microtubules. Hence, that's why its name is intermediate filaments. So as I told that the microtubules were formed by the tubulin protein, and the subunit protein of the intermediate filament is vimentin. As you can see over here, vimentin or vimentin, how you pronunciate it. And these vimentin also form the chains like this. And these chains, when these chains are three, when vimentin molecules make three chains, it results in the formation of intermediate filament. Here is the one, here is the two, and in between there is running the third one as well. Look, one, two, and three. And these filaments are twisted in such a way that there is no hollow space, such as that was in microtubules. So what's the function of intermediate filaments? They basically provide the mechanical support to the nuclear envelope and plasma membrane. So that was all about the cytoskeleton, which makes up the microfilaments, microtubules, intermediate filaments. Hope you got the concept. Now let's move on to the brainstorming session. Filaments present in flagella and cilia are microfilaments, microtubules, microvilli, intermediate filaments. For the answers, you may listen the lecture carefully and then come on to this. So the right answer is microtubules. Let's move on to the next one. Diameter of the microfilaments is 8 nanometer, 10 nanometers, 7 nanometers, 6 nanometers. The answer is simple. It is 7 nanometer. So the next one, it provides mechanical support, microfilaments, intermediate filaments, microtubules, microvilli. So the answer is, it's easy, intermediate filaments. So that was all about the today's lecture. Stay tuned for the upcoming lectures and show some love by liking the video. Thank you.